Glucose is the most important sugar in the human body. It is found in numerous foods and has several common names, including dextrose, grape sugar, and blood sugar. Glucose is broken down in glycolysis and other pathways to release energy for body functions. The concentration of glucose in the blood is critical to normal body function. As a result, it is carefully controlled by the hormones insulin and glucagon. Normal blood glucose levels should be under 100 milligrams of glucose per 100 milliliters of blood, with the highest concentrations appearing after a meal. Insulin stimulates the uptake of excess glucose by most cells of the body, and after 1 to 2 hours, the levels return to normal. If blood glucose concentrations drop too low, the individual feels lightheaded or shaky. When this happens, glucagon stimulates the liver to release glucose into the blood, re-establishing normal levels. We're going to take a closer look at this delicate balancing act in Chapter 23. The molecular formula of glucose and aldohexose is C6H12O6. What is known as the open chain form of glucose is shown here. And what's interesting is that glucose can not only exist in an open chain form, but it can also exist in one of two cyclic forms. We can have what is called alpha D-glucose and what is called beta D-glucose. And the way that alpha and beta D-glucose are formed is by a reaction between the lone pair on the hydroxyl of carbon 5 and the carbonyl. During the process, carbon 1, shown here, becomes a chiral center. And the chiral center, or carbon 1, is called the anomeric carbon or the anomeric center. When the hydroxyl at carbon 1 is pointing down, we call that the alpha anomer. And when the hydroxyl at carbon 1 is pointing up, we call that the beta anomer. So the word that we use is, instead of isomer, we call it an anomer because it's an isomer on an anomeric center, right? This is the anomeric carbon and this is the anomeric carbon, or we can call it the anomeric center. And my students taught me an easy way to remember alpha and beta. They said that alpha looks like a fish, and fish live underneath the water, so that's why the hydroxyl is down for an alpha. And beta kind of looks like a butterfly, and butterflies live up in the air. So that's why the beta hydroxyl is the one pointing up. Let me show you how to draw the cyclic forms, starting with the open chain form. And the way that we do that is we start with our open chain form here. So here's our D-glucose molecule, and then we pretend we're chopping it down like a tree and that it's going to fall to the right. So if we were to knock this over, it says here it's rotating it, but if you think about knocking it over, if it was to fall on the ground like that, what would it look like? It would look like what we see here in B, where carbon number 1 is way over here, carbon number 6 is going to be way at the bottom like this. So any of the groups that are on the right-hand side of the molecule are the ones that are going to be down on the ground like that. If you don't believe me, look, we have a hydroxyl, a hydroxyl, a hydrogen, a hydrogen, another hydroxyl, another hydroxyl, another hydroxyl, and another hydroxyl. The next part involves a little memorization, and I'm just going to coach you on how to draw this part here. So the first thing you do is you draw a hexagon, but you leave out the top right line like that. You put the most oxidized carbon, or the aldehyde in this case, over here on the far right. Then you put a hydroxyl at this point right here, and then the last thing you have to memorize is to put a CH2OH up here. Next, we're going to number the carbons the same way that they're numbered, shown here. And so we'll start with the aldehyde carbon, 1, 2, 3, 4, 5, and 6. These are the same carbons that are numbered here, 1, 2, 3, 4, 5, 6. Anything that's pointing down like this is going to be pointing down on our new structure that we're drawing. And on carbon 2, we have a hydroxyl pointing down, so we'll put the hydroxyl pointing down. On carbon 3, we have a hydrogen pointing down. On carbon 4, we have a hydroxyl pointing down. And the hydroxyl on carbon 5, that's this one shown right here. And you can include this hydrogen that I've highlighted in green pointing down like this. And then on carbons 2, 3, and 4, you basically fill in what's left. So on carbon 2, if we have a hydroxyl pointing down, that means we have a hydrogen pointing up. On carbon 3, if we have a hydrogen pointing down, that means we have a hydroxyl going up. And on carbon 4, if the hydroxyl is down, the proton is up, or the hydrogen is up. And those are these 3 atoms that you see right here, right? This hydrogen is pointed up on carbon 2, the hydroxyl is up on carbon 3, and the hydrogen is up on carbon 4. Next, we can draw an arrow for the reaction that takes place to make the cyclic form. And so one of the lone pairs is going to form a bond with this carbon right here on the aldehyde. And when that happens, we get something that looks like this. The next part is just to decide whether we're going to draw alpha D-glucose or beta D-glucose. If we want to draw alpha D-glucose, the hydroxyl should be pointing down like this. And if we want to draw beta D-glucose, the hydroxyl should be pointing up like this. In actuality, the open chain form of glucose is present in very small concentrations in cells. It exists mostly as the cyclic form under physiological conditions because the carbonyl group at carbon 1 of glucose reacts with that hydroxyl group at carbon 5 to give a 6-membered ring like these that are shown here. In the discussion of aldehydes, we noted that the reaction between an aldehyde and an alcohol gives us a hemiacetal. When the carbonyl group of the aldehyde portion of the glucose molecule reacts with the carbon 5 hydroxyl group, the product is an intramolecular hemiacetal. Let me show you the hemiacetals in both alpha and beta D-glucose. They go like this. We have a carbon, then an oxygen, then another carbon, then an oxygen, and a hydrogen. That is a hemiacetal. The same thing applies right here. Carbon, oxygen, hydrogen. If you need a refresher of the hemiacetal, what I told you before is that when you have a carbon attached to a hydroxyl and an ether, it's considered a hemiacetal. These representations that we see here are called Haworth projections. Sometimes they're pronounced Haworth projections. Here's another example showing you how to draw a Haworth projection or the cyclic form of D-mannose, which is shown here as a Fisher projection. This is the Dr. Deniston method, or the method devised by the author of our textbook. So what she says here is, in this example, we're going to draw both anomers. So that means we're going to draw both the alpha and beta anomers of this molecule down here, D-mannose, in a Haworth projection. And the way that she likes to do it is to get her students to memorize this part right here, because this is common in all D sugars. So if you can remember the 6-membered ring with the oxygen in the top right corner and the CH2OH right next to it above the plane of the 6-membered ring, filling in the rest is pretty easy. And the way you would do that is to start by numbering the carbon. So we have carbon 1, 2, 3, 4, 5, and 6. We'll number them over here. Here's our anomeric carbon, 1, 2, 3, 4, 5, and 6. Remember that on the carbons 2, 3, and 4, so carbons 2, 3, and 4, anything that's pointing on the right-hand side is going to be pointing down. So that means on carbon 2, I have a hydrogen going down. If I have a hydrogen going down, that means the hydroxyl is going up. On carbon 3, I also have a hydrogen pointing down, therefore I have a hydroxyl pointing up like this. And on carbon 4, my hydroxyl is pointing to the right, so therefore it must be pointing down, and therefore I have a hydrogen up like this. The next part is deciding whether we're going to draw alpha D-mannose or beta D-mannose. Let's start by drawing alpha D-mannose, and remember that when we have alpha, our hydroxyl at the anomeric carbon is going to be pointing down like this. Therefore, this is alpha D-mannose. If I want to draw the beta anomer, beta D-mannose, remember that the hydroxyl in the beta anomer is going to be pointing up like this, and so this would be beta D-mannose. It's important to remember that the only way that alpha and beta D-mannose differ is the orientation of the hydroxyl group at the chiral carbon. And here's her drawings of the two anomers, the alpha anomer that shows the hydroxyl on the anomeric carbon pointing down, and the beta anomer which shows that hydroxyl pointing up like this. Fructose, also called levulose or fruit sugar, is the sweetest of all sugars. It is found in large amounts in honey, corn syrup, and sweet fruits. The structure of fructose is similar to that of glucose. When there is a CH2OH group instead of a CHO or an aldehyde group at carbon 1 and a carbonyl group instead of a hydroxyl at carbon 2, the sugar is a ketose, in this case D-fructose. Fructose, also called levulose or fruit sugar, is the sweetest of all sugars. It is found in large amounts in honey, corn syrup, and sweet fruits. The structure of fructose is similar to that of glucose. In fact, they have the same molecular formula. But you can see that fructose is in fact a ketose and not an aldose. Since it has 6 total carbons, we call it a ketohexose. Cyclization of fructose produces alpha and beta D-fructose, shown over here. In both alpha D-fructose and beta D-fructose, shown in this slide, the hemiacetal is highlighted. Fructose forms a 5-membered ring structure. You will not be asked to draw the cyclic structure of fructose from the Fischer projection. Another important aldohexose is galactose. It is a diastereomer of glucose. The linear structure of D-galactose and the Haworth projections of alpha D-galactose and beta D-galactose are shown on this slide. Galactose is found in biological systems as a component of the disaccharide lactose or milk sugar. This is the principal sugar found in the milk of most mammals. Beta D-galactose and a modified form called beta-D-N-acetylgalactosamine are also components of the blood group antigens. You can see that the only difference between beta-D-glucose and beta-D-galactose is the orientation of the hydroxyl group at carbon 4. In glucose, it's pointing down, and in galactose, it's pointing up. Ribose, an aldopentose, is a component of many biologically important molecules, including RNA and various coenzymes that are required by many of the enzymes that carry out biochemical reactions in the body. The structure of the 5-carbon sugar D-ribose is shown here. DNA, the molecule that carries the genetic information of the cell, contains an aldopentose beta-D2-deoxyribose. In this molecule, the hydroxyl group found in ribose at carbon 2 has been replaced by a hydrogen. For this reason, this monosaccharide is called 2-deoxy, indicating the absence of oxygen. The aldehyde group of aldoses is readily oxidized by the Benedict's reagent. Recall that the Benedict's reagent is a basic buffer solution that contains copper-2 ions. The copper-2 ions are reduced to copper-1 ions, which, in basic solution, precipitate as brick red copper-1 oxide. The aldehyde group of the aldose is oxidized to a carboxylic acid, which undergoes an acid-base reaction to produce a carboxylate anion. And you can see here that the aldehyde is being oxidized to the carboxylate. Why? Because it's losing hydrogen and gaining oxygen. But in the process, the copper-2 is gaining electrons to form copper-1. This is copper-1 and copper-1 oxide. And so the copper is being reduced. And so any sugar that can reduce copper-2 to copper-1 is called a reducing sugar. All monosaccharides and the disaccharides that we'll look at, except sucrose, are reducing sugars. So this is a point you should memorize, that all mono and disaccharides, except for sucrose, table sugar, are reducing sugars. Although ketones are generally not easily oxidized, ketoses are an exception to that rule. Because of the hydroxyl group on the carbon next to the carbonyl group, ketoses can be converted to aldoses under basic conditions via an ene-diol reaction. The name of the ene-diol reaction is derived from the structure of the intermediate through which the ketose is converted to an aldose. It has a double bond, ene, and 2 hydroxyl groups, and that's where diol comes from. Because of this ene-diol reaction, ketoses are also able to react with Benedict's reagent, which is basic. Because the metal ions in the solution are reduced, the sugars are serving as reducing agents and are called reducing sugars. Again, all monosaccharides and all the common disaccharides except sucrose are reducing sugars. For many years, the Benedict's reagent was used to test for glucosuria, the presence of excess glucose in the urine.